Hello everyone, welcome back to my channel Bio Universe. I am Suyanka. From today, we will start revising some chapters of class 9 syllabus. First chapter we will revise is the fundamental unit of life. Before that, please subscribe my channel. In 1665, Robert Hooke discovered that a thin slice of cork from bark of a tree contained a number of small boxes. He termed it cellula, which means little room. This discovery was important because it indicated for the first time that living organisms consisted of a number of smaller structures or units. As we all know that buildings is made up of bricks. Similarly, the bodies of all plants and animals are made up of cells. We can define cells as the fundamental structural units of living organisms and the basic unit of life. The study of cells are known as cell biology. Discovery of cell. In 1665, Robert Hooke observed honeycomb-like structure and termed cellula, which means little rooms. He published his work in book Micrographia. In 1674, A. V. Leeuwenhoek, a Dutch scientist, studied living cells like bacteria, protozoa, RBC, etc. for the first time. In 1831, a Scottish botanist, Robert Brown, discovered and named the nucleus in a cell. In 1839, J. E. Purkinje used the term protoplasm for the living substances present inside the cell. In 1839, two German biologists, Swan and Sladen, formulated cell theory. According to them, the cell is the structural and functional unit of all living beings. In 1855, Roger Virchow, a German pathologist, stated omnis cellula e cellula, which means all cells arises from a pre-existing cells. He modified the cell theory by adding this generalization. All living beings show cellular organization. However, there are some organisms like amoeba, paramecium, bacteria, etc. whose bodies are made up of one cells. They are known as unicellular organisms. The organisms which are made up of a number of cells are called multicellular organisms. For example, fungi, plants, and animals. Shape and size of cells. The size and shape of cells are not similar in all organisms. It can be related to the specific function they perform. Cells can be polygonal like liver cells, spherical like ovum, etc. Some cells can change their shapes like amoeba, while some of the cells have fixed shapes like plant cells and animal cells. Some of the cells can be seen with naked eyes, but a great majority of cells are too small to be seen with the naked eyes. We can see these cells through microscopes. Size of mycoplasma is 0.1 to 0.5 micrometer. Human RBC is 7 micrometer in diameter. Human liver and kidney cell is 20 to 30 micrometer. Nerve cell is 90 to 100 centimeter. Ostrich egg is 18 centimeter. Smallest cell of the world is PPLO, mycoplasma. Largest cell is ostrich egg. Longest cell is nerve cell. What is the structural organization of a cell? If we study a cell under a microscope, we will observe a cell as a teeny mass of protoplasm which is limited by a membrane and capable of performing all functions of life. A typical cell is formed of three parts: plasma membrane, nucleus, and cytoplasm. Let us see them one by one. First is plasma membrane, also known as cell membrane. The outermost covering of the cell that separates the contents of the cell from its external environment is called plasma membrane. Plasma membrane is selectively permeable because it allows entry of certain substances, exit of some substances while preventing passage to remaining substances. Plasma membrane is a living flexible membrane. It is 6 to 8 nanometer in thickness. Plasma membrane is made up of lipid bilayer, proteins and carbohydrates. Functions of plasma membrane. It provides a definite shape to semi-fluid contents of the cell. It functions as a mechanical barrier that protects the internal contents of the cells. The membrane determines what substances are to be allowed entry into or exit from the cell. The flexibility of the membrane enables the cell to ingest the food and other substances from its external environment by endocytosis. It has chemicals which help cell recognition for tissue formation, distinction of foreign substances, and defense against microbes. It provides for flow of information among different cells of the same organism. Transport across the membrane. Membranes act as physical barriers between the organelles of a cell and the cytoplasm, and between the cell and its surrounding environment. The membrane is said to be impermeable if the substances do not pass through the membrane. Permeable if the substances, both solute and solvent, can pass readily through the membrane. Semi-permeable if the membrane is permeable to solvent but prevents the passage of solutes. Selectively permeable if the membrane allows the passage of solvent and some selected solutes. There are two types of transport mechanisms, active and passive. Active transport. It is a type of transport across plasma membrane which requires the use of energy. It usually occurs against the concentration gradient. For active transport, the cell membranes possesses carrier proteins. Glucose, amino acids and some ions pass through the plasma membrane by an active process. Passive transport. It is the passage of substances across the plasma membrane without expenditure of energy. It is of two types, diffusion and osmosis. Diffusion, the process of movement of a substances from a region of a higher concentration to a region of lower concentration to spread uniformly in the given space, is called diffusion. Osmosis. Osmosis can be defined as the diffusion of water or solvent across a semi-permeable membrane from a region of higher concentration to the region of its lower concentration. Types of osmotic solutions. Osmotic solutions are those solutions which can cause osmosis if separated from solvent by semi-permeable membrane. They are of three types: hypotonic, isotonic and hypertonic. Hypotonic solutions is a dilute solution which has an osmotic concentration lower than that of a other solution. Isotonic solution are the solution has an osmotic concentration similar to that of another solution. Hypertonic solution is the concentrated solution which has an osmotic concentration higher than that of the another solution. Types of osmosis. Osmosis is of two types, endosmosis and exosmosis. Endosmosis is the osmotic entry of water into a cell or system. Exosmosis is the osmotic withdrawal of the water from a cell or system. What will happen if we put an animal cell or a plant cell into a solution of a sugar or a salt in water? One of the following three things could happen. If we put a cell into a hypotonic solution, water molecule passes through it in both directions, from outside to inside and from inside to outside. However, more water molecules will come into cell than leave. It is due to difference in their concentration. The net result is that water enters the cell wall will swell. This phenomenon is called endosmosis. If we put the cell into isotonic solution, water passes the semi-permeable membrane in both directions. The amount of water going inside the cell is equal to the amount of water going out of the cell. Therefore, there is no net movement of water. The cell size will remain the same. If we put the cell in hypertonic solution, more water concentration is present inside the cell. Plasma membrane, which is semi-permeable membrane, allows movement of water both into outside solution from the cell as well as into the cell from the outside. However, since more water is present inside the cell, more of it will pass out. The phenomenon is called exosmosis. As a result, the cell wall will shrink and plant cells, the shrinking protoplasma, will contract from the cell wall. It is called plasmolysis. Cell wall. Cell wall is a rigid, semi-elastic, semi-transparent, supportive and protective covering present outside the plasma membrane in plant cells, fungi, some protists and prokaryotes. They are mainly made up of cellulose. Cellulose is a complex fibrous carbohydrate which cannot be digested by human beings. It is the most abundant organic molecule. Cell wall possesses many small pores through which adjacent cells remain connected. These pores, having cytoplasmic bridges between adjacent cells, are called plasmodesmata. Functions of cell wall. Cell wall provides shape to the cells. It provides mechanical strength to plants. Cell wall protects the cells against pathogens and mechanical injury. Cell wall prevents bursting of cell on endosmosis. Because of cell walls, plant cells can withstand a lot of variations in the surrounding medium as compared to animal cells. That's all for today. We will discuss remaining chapters in the next video. Thank you.